Ever wonder how your cells keep from swelling up and bursting? Or, how they stay charged and ready to fire at a millisecond's notice? Well, today we are unpacking some absolute foundational material from the AllAboutRx notes. And really, it all comes down to two main things: the resting membrane potential and the absolute unsung hero of your biology, the sodium-potassium pump. So to kick things off, why doesn't your cell explode? It is a serious biological threat. If you don't have a way to manage the ions inside and outside the cell, water rushes in and boom. The answer to this literal life or death situation lies in understanding our cellular baseline and the incredible little engine that maintains it. Okay, let's dive into this. Let's talk about that baseline, the resting membrane potential. Before a neuron can do any active work, it has this resting electrical state. It's essentially the difference in voltage between the inside and the outside of the cell membrane. To really get how the pump works, we have to understand this environment first. And honestly, it's an environment so critical that your brain spends up to half of its total energy just maintaining it. Check this out. Notice that sage green highlighting the potassium leak channel against the beige background there? At rest, your cell membrane is super permeable to potassium. Since there's a high concentration of potassium inside the cell, it naturally just leaks out down its concentration gradient. But look at what it leaves behind in the cytoplasm, those A- blocks. Those are trapped intracellular anions. We're talking heavy proteins and phosphates that literally just cannot cross the membrane. So positive charge leaves and negative charge gets trapped inside. And that dynamic gives us a crucial number to cement in your brain for your board exams, roughly -70 millivolts. That is your baseline. Now, remember the sign convention here. That negative sign doesn't mean the voltage is literally less than 0, right? It simply means the cell interior is more negative relative to the outside fluid. The outward leak of potassium eventually gets balanced out by the inward electrical pull of those trapped negative proteins settling right at that -70 baseline. But here is the catch. Ions constantly leak. If we just left it unchecked, the cell would eventually lose its gradients entirely. The battery would just die. So to stop that gradient from dissipating, we need an engine. Enter the Sodium-Potassium Pump, or Na K ATPase. This is a primary active transport mechanism, meaning it directly burns ATP to shove ions against their natural flow. Now, how does this engine actually work? It runs on a continuous 6-step catalytic cycle. The pump itself is mostly made of this large transmembrane alpha subunit. That's the catalytic part doing the actual heavy lifting, along with a beta regulatory subunit. By burning ATP, the pump literally physically changes shape back and forth between two main structural states, called E1 and E2. Grabbing ions on one side and chucking them out the other. Let's break down that loop real quick. Step 1. The pump is open to the inside, that's the E1 state, and it binds 3 sodium ions. Step 2. ATP gets hydrolyzed, leaving a high-energy phosphate attached to the pump. This phosphorylation triggers step 3, which is a massive shape shift to the E2 state, dumping those 3 sodiums outside. Now, step 4. This new E2 shape absolutely loves potassium, so it grabs 2 K+ ions from the outside. Step 5, the phosphate pops off, and finally, step 6, dephosphorylation snaps the pump right back to its original E1 shape, dropping the 2 potassiums inside. The super easy mnemonic here for your exams, the rule of 3 and 2. 3 sodium out, 2 potassium in. Now, what's really interesting about this is the math. Think about it. We're pumping 3 positive charges out, but only bringing 2 positive charges in. So we have a net loss of 1 positive charge for every single cycle. This makes the pump electrogenic. It isn't just maintaining gradients. It's actively contributing a few millivolts directly to that negative resting membrane potential we just talked about. I want to pause deliberately for a second here. Let that basic mechanism really sink in. 3 sodium out, 2 potassium in, 1 ATP burned. Got it? Awesome. Because we're about to transition from basic physiology to high-stakes pharmacology. How do we take this exact machine and manipulate it to save patients? Well, we can literally turn this pump down or crank it up. Take a look at the left side. We've got digoxin, a classic drug used in heart failure. Digoxin binds to the outside of that alpha subunit and inhibits it. Why would we do that? Well, if the pump stops, sodium builds up inside the heart cell. That knocks out a secondary transporter, the sodium-calcium exchanger, which causes calcium to build up inside the cell. And more calcium means a stronger, harder heartbeat. But then look at the right side. In an emergency like hyperkalemia, where blood potassium is dangerously high, we use IV insulin and glucose.
Insulin aggressively stimulates this exact same pump, forcing it into overdrive to pull that dangerous potassium out of the blood and hide it safely inside the cells. It's wild, right? Two totally opposite clinical interventions, manipulating the exact same biological engine.
And this brings us right back to our opening hook, the threat of pump failure. What happens when this goes wrong unintentionally? If your tissues suffer from hypoxia or ischemia, ATP production just stops. Without ATP, the pump fails. Sodium rapidly accumulates inside the cell. And remember your basic biology. Wherever sodium goes, water follows osmotically. The cell draws in massive amounts of water, swells up, and ultimately lyses. It explodes, permanently destroying the tissue. Okay, as we wrap up this explainer, let's run through a fast-paced, quick recall of the absolute highest yield board facts here. Ready? Resting membrane potential is about -70 millivolts, mostly set by potassium leaking out. Pump stoichiometry, 3 sodium out, 2 potassium in, 1 ATP burned. It's a P-type ATPase with an active alpha subunit. You can stimulate it with insulin, thyroid hormone, and aldosterone. You inhibit it with digoxin or hypoxia. Digoxin boosts cardiac contractility via that Na plus C2 plus exchanger, and insulin is your rapid emergency fix to shift potassium intracellularly. The Na K pump truly is the engine of cellular life. It is an absolute necessity. I mean, using up to 70% of a neuron's ATP just to maintain readiness, that is a massive energetic commitment to keep your cells from exploding. Think about what that implies for the ischemic cascade the very second blood flow stops. To dive deeper into that and to explore the rest of these clinical pearls, definitely check out the full AllAboutRx study notes. Stay curious, stay informed, and I'll catch you next time.